Yeah, we didn't come up with that. There's always this element of serendipity when you start a company. It was Bristol Myers Squibb and AstraZeneca almost independently that saw the instrument. It was originally pitched for a different application.
That's Frank Feist, CEO of BlueCatBio, a trailblazer in lab automation. From their signature blue washer to tools including BluePlate, BlueBench, and BlueCatX, BlueCatBio is redefining how researchers can enhance their cellular workflows with flexibility, throughput, and reproducibility. Welcome to the Biotech Stack, where we explore the cutting-edge technology fueling the future of life sciences and biotech. From next-generation lab automation and AI-driven drug discovery to breakthrough biomanufacturing and research tools, we dive deep into the innovations shaping the next wave of blockbuster medicines and the businesses behind them. I'm Corinne Regan, your host. In today's episode of the Biotech Stack, we talk to Frank Feist, CEO of BlueCatBio. We hear about Frank's journey from going from telecommunications engineer to the CEO of BlueCatBio. We chat about the ways BlueCatBio is helping shape the life sciences research industry and what the future of the company is. Let's get started. Okay, Frank, so I am going to jump right into things, and I would love it if you could share a little bit with our audience who you are and a bit about your background.
Well, our company is BlueCatBio. We started BlueCatBio 11 years ago. It was two partners, two very good friends in Germany, myself. We started BlueCatBio simultaneously in Germany and in the US, and that was really critical for making the company a success because we have very good engineering and manufacturing capabilities in Germany, very attractive markets in the US, and so it pretty much took off right from the beginning. Myself, I'm trained as an electrical engineer. I got trained in biology by my two partners, who are both PhDs in biology, and I have a business degree. So even though my background was originally in telecommunications, I've been in the life sciences now for over 20 years, and it's just a field I enjoy tremendously.
That's interesting, an interesting background. So how, if you were an engineer in this space, did you connect with your partners who were in a totally different space, who are biologists, it sounds like?
Yeah, when I met them, I had already done the migration from engineering into life sciences business development. That was from a previous company where we met. So they never met me as a telecoms engineer. Having said that, I was sort of tongue-in-cheek, like to joke about that. The commonality between telecommunications and sort of high-throughput biology is signal-to-noise. At the end of the day, it's a lot of data, and you have to separate the background from the actual signal that you're trying to detect, and that is exactly the same challenge in telecoms and in biology.
Interesting. That's an interesting take. I like that. And then you did mention that the company started one in Germany, one in here in the US. Are they separate entities or are they the same company?
We are legally separate entities, and for taxes and finances and everything, we're completely separate, but we have the same ownership. And we operate under, BlueCatBio as a global brand. Yeah, there's no distinction based on market. We have the same marketing material and everything, but we're legally separate.
Gotcha. So to dive a little bit more into BlueCatBio itself, could you explain to us, the audience here, a little bit more about BlueCatBio and what you do?
Yeah, so we make instruments for mostly pharmaceutical companies. So we are a provider of innovative liquid handling and laboratory automation solutions. Our original product is the Blue Washer, which is a new take on washing plates. Removing liquid not by aspiration like it's been conventionally done, but using centrifugation to evacuate plates, which has the advantage that the evacuation is much more stringent. There's much less residual, and it's also much more reproducible. And in the context of cellular assays, that translates into better data quality that the customers get at the end of the assay. So we won the New Product Award at SLAS 2015 for that product. We won the New Product Award again in 2024 for our BluePlate product, which is translating the concept of centrifugal evacuation into flow cytometry and applying it to washing cells suspension cells, as well as cells that naturally adhere to the bottom of a plate. And we just launched our new product, which is now able to fully automate a flow cytometry workflow.
Oh, that's great. Can you go into a little more detail on that new product?
Yes. So the success of BlueCatBio was really that we gave a means to, or a method to pharmaceutical companies that dramatically improves the data quality of their cell-based assays just by using centrifugal evacuation. And customers typically work with both adherent cellular assays as well as suspension assays in their screening applications. So they asked us, well, don't you have a solution to provide the same sort of lift in data quality for suspension cells? And we thought long and hard about that issue because it's a difficult one. Flow cytometry sample prep is one of the few remaining workflows, I would say, for which there's very few automation solutions that really check all the boxes. It's, for most part, still a very labor-intensive manual process where lab techs would take a sample, spin it down into a pellet. It takes a couple of minutes, requires a big centrifuge, then pipetting off the supernatant, and then repeating that many times before they can put the prepared sample onto a flow cytometer. Our approach is using the blue plate as a consumable, which provides for the separation between the solid and the liquid, and then our new instrument, the BlueCombi X, which both does the settlement of the cells into the bottom part of the blue plate, as well as the evacuation. So it's a full walk-away automation of the flow cytometry step.
So diving in a little bit more, based on these different products you offer, what would be some of your ideal customers and what market does this fit into?
Yeah, so to date, I would say about 90% of our customers are pharmaceutical companies that use our products mostly for drug discovery workflows in cellular screening. Having said that, there's a good amount of customers in diagnostics that use the instrument often for magnetic bead-based workflows where magnetic beads need to be washed, and that can be done on our blue washer and the new instrument as well.
And so with these products and being a global company, what does your team look like, those who are supporting customers out there in these pharma companies and diagnostic companies?
Right. Yeah, we have 5 people in the field in the US and about the same amount in Europe. And I always like to say our people, they wear 5 hats, which from the customer experience makes it very different from big companies. So our customers typically deal with the same person from when they get quotes to when they get a demo, to when they get the instrument installed, to when they have questions about assay development and support. Same person would also deliver an annual preventive maintenance service, fix anything if anything needs fixing. Yep.
That's great. I love that model of having kind of one point of contact who can kind of do it all. That's great.
Exactly. Yep.
So we like to ask this question about, your product and offerings. Is there anything different about your product compared to others in the market? Perhaps there's a secret sauce to how you've built things.
Yeah, so the concept of centrifugal evacuation was completely new when we came onto the scene. So that really is the innovation. Before the blue washer, pharmaceutical companies would use conventional aspiration washers or liquid handlers to remove the liquid from a cell-based assay. Typically, the first removal step is right after a cell comes out of the incubator in proliferation. They have growth medium that's part of, that makes the cells grow overnight. That growth medium needs to be removed. And because growth medium contains sugar, which is very reactive and would mask the effect of the pharmaceutical compounds that they want to test, typically used to require multiple PBS wash steps to really get all the residual sugar components out. With a blue washer, that basically works in one step. And because our residual volume is typically only half a microliter or less per well, and that compares to about 10 or 20 microliters that you would have in the conventional workflow, the completeness of removal of medium is really what drives the downstream data quality.
Gotcha. So if this is such a new concept, this centrifugation in this workflow, what led you and your team to thinking that they can use a centrifuge process to do this?
Yeah, we didn't come up with that. It was, there's always this element of serendipity when you start a company. It was actually, I think, Bristol Myers Squibb and AstraZeneca almost independently that saw the instrument. It was originally pitched for a different application. It was originally invented to do 384-well ELISA without having to go through many wash steps. But they found out about the instrument, and it was through the customers that we learned that the completeness of media removal was very important to them. And literally my first demo that I ran at BMS here in the US, the customer had, big old Beckman centrifuge and napkins from the cafeteria. So you would stick the napkins on top of the plate, then spin it so that the napkins would absorb the media and say, look at this, I have to automate this. What can I do? And that's how we discovered that application and the fact that it was really important to customers. And then there was other customers like Mary Jo Wildy at Merck, who discovered that the Z prime, which is a parameter that basically characterizes the statistical reliability of an assay, that it was improved tremendously by this approach. And then, the rest is history.
Oh, that's very interesting. That's a cool way of developing a product, going from one concept to another based on customer experience, right? That's the best way to do it. Speaking of Merck, there is a great story between BlueCatBio and Merck and how maybe your technology helped solve a very critical problem that they have. I'm sure you've told this story before, but I would love it if you could share that story with us again.
Yeah, it was a great story, and we are eternally grateful to Mary Jo for finding it out and then being persistent to sort of spread that insight within the Merck organization. But long story short, she had a number of assays that were borderline in terms of Z prime, which for them is a 0.5 Z prime value. They need to hit 0.6 or better to operationalize an assay. And with conventional washers, she just wasn't able to hit that mark. And once she ran the blue washer, the Z prime immediately improved. And they even found out that one of the compounds that they had in their library and have been testing in their screens for 30 plus years had never shown any activity. And then all of a sudden, with the blue washer, it showed activity. And the biochemists and bioinformaticians who, sat on the back end of the data analysis, they were extremely skeptical at first. They said, it's just a bad instrument. But then all the dose responses and the secondary assays they checked out, the compound remained active. And what they then figured out was that it was the sugar in the growth medium that had not been sufficiently removed in previous methods that basically masked the innate activity potential of that compound. And it was really that insight that drove Merck to fully adopt the blue washer method across their screening.
That's so interesting. That brings me to a question, is there seems to be limiting factors in the scientific discovery, but it's complicated, right? So is that scientific discovery being limited by the technology that's available? Is it being limited by the ability for scientists to uncover new things? In this case, it sounds like technology limited the ability for true science to further progress. Do you still see that as maybe some of the limiting factors that our technology isn't advanced enough yet? Or are you starting to see the scientific advancements and knowledge catch up as well?
Well, that's sort of a big question.
Yeah, it's a big question.
I think maybe my sort of insight from this is that a lot in the overall improvement of the drug discovery workflow, the capital efficiency of the workflow, start to finish. Basically, you start with a concept, and then you need to create target compounds that then are evaluated further in the drug discovery chain. A lot of the effort has been placed on post-wet lab data processing mostly on the image side. So, there's the optical microscopes and automatic scanners that are now being used are just incredibly sophisticated, and they layer multiple images on top of each other and use those to cancel out noise. But I think our twist on that is from a capital effectiveness perspective. You can achieve a lot more data improvement by removing the background where you make it, which is in the wet assay. And that was just a different tack, that we took, a different way to solve the problem. And of course, the imaging is super important as well. But taking both of those things together, I think that made a real step forward in drug discovery efficiency.
Gotcha. Going back a little bit more to BlueCatBio's technology itself. So you are involved in high throughput screening, you are involved in automation. We've had experience that there is a little bit of apprehension when you're moving folks in the lab to these automated processes. Do you experience that with your clients? If so, do you have a method with your team to overcome some of the hurdles and the apprehension in that automated transition?
When you say apprehension, do you mean in terms of automation being a job killer, or?
That's I didn't think of it that way, but that is also something we can touch on as well. I thought of it more as there is apprehension in our world to take ourselves out of the equation from a manual perspective. While manual processes can lead to inconsistencies across the board, you also feel like you have control over how your workflow and your scientific assay is going to come to fruition. But automation is now taking the inconsistencies out. They're helping to make things faster. But there is still that human interaction where there's apprehension to want to move in that direction. And then, like you just mentioned, there's also that apprehension, but if I lean on automation, will I then take my own job away by teaching it how to do my workflow? So maybe you can tackle both. How do you see it being in this world of automation?
Yeah, I mean, the human touch, I think what we see typically when we work with our customers, our instruments are, when you look at our global installed base, it's pretty much a 50-50 split between instruments used in a benchtop setting by a scientist who typically would either just run a very small screen or develop a method which then gets automated. And then the other half is fully integrated and automated with any of the vendors of such equipment. So I think the human touch really comes in when assays are developed. And there's a lot of, literally hands-on work, running it, looking at the result, optimizing it, tweaking it. I think the advantage of our instrument is that the step from manually running something using the blue washer to then automating is not very big. Because as opposed to conventional washers, or, when you take a plate and you dump and tap it, right, which is the sort of the old-fashioned way to get liquid out of a plate, taking an SOP that has manual tapping it and then automating it, that's a big step. Having an SOP that you developed on the benchtop on the blue washer, which actually includes writing a program with all the settings that run inside the blue washer, and then transferring that over to an automated platform, you really don't lose any kind of the performance characteristics of the assay. So I think the scientists feel very comfortable that what they have developed manually on their benchtop, once they transfer it over to automation, it delivers the exact same results.
Gotcha. Okay. So having a workflow that's able to scale from this manual, more benchtop process to a fully automated process, you think has helped clients and customers to kind of overcome that much bigger hurdle of just jumping right in from manual to automation.
Exactly. Yeah. And, back to Merck, one of the big drivers of their adoption was also a realization that with conventional washers, there was a lot of site-to-site and operator-to-operator variability in results, just because it's a complex machine. And the fact that you have to adjust the needle, how deep do you put the needle? You want to keep it as low as possible to minimize residual volume. You don't want to get too low because then you can suck up cells. Those are all individual judgment calls that are very difficult to operationalize and to standardize. And in the Blue Washer, it's literally you develop the protocol, you write the script for it, and then you just email it to anybody else who wants to run it, and they get the same results.
Gotcha. So you've talked a little bit about some of the applications that BlueCatBio supports, right? Cell washing. You've talked a little bit about sample prep for flow cytometry. I've seen that you guys also have application support for the non-tip magnetic bead separation. Are there any other applications that your team is focusing on at the moment? Maybe some innovations that BlueCatBio will see in maybe the next 3 to 5 years?
Yeah, I mean, a big topic is always 3D cellular assays. 3D cellular assays are very appealing because their results tend to correlate more with down the road studies, like mouse studies, for instance, than just plain 2D assays or even flow assays. So we are extremely interested in that as well. And we have applications for it both on the conventional Blue Washer, but also now in the Blue Plate, because the same concept of having this, what we call the cell-safe compartment, where we retain the suspension cells so they don't get ejected during the centrifugal step. The same physics can be used to safely contain a 3D organoid, for instance, down there, and then do a media change on top. So yeah, that's another application that is extremely exciting for us.
Okay. Is somewhere on the roadmap in the future, we have something already developed for that application currently?
Yeah, we already have customers who use it for 3D. In terms of the Blue Plate, we are going to have versions of the Blue Plate that are very transparent from an optical perspective, because that's important for the downstream data acquisition of an organoid. And there will be more very specific products for that application coming to market.
Gotcha. Very cool. I'd like to go a little bit bigger picture here. I know that we got an opportunity to see each other at SLAS in San Diego, and a lot of things have changed macroeconomically. We've had some changes with NIH losing funding. The FDA has faced some challenges. There are obviously tariffs. You're being a global company. Has BlueCatBio experienced any changes since, last time we had a chance to see each other?
Yeah, I mean, tariffs is an easy one. We import from Germany, so we're now subject to a 15% import tax. I mean, the way I look at that is we have to focus on providing extremely high value products to our customers that allow us to have sufficient margin to absorb the import tax, or to some extent pass it on. I would say it's not a tremendous factor for us, even though it does cut into our margin for now. But our response is just to really focus on the value that we create, and in particular, our new instrument is it's the world's first laboratory instruments that's both a centrifuge and a washer, and we price it such that it's about 20% cheaper than two separate instruments. So right there's an opportunity for us to really create value for the customers that, at the end of the day, is much more important than some increase in input cost for us.
Well, that's good news. It's good news to hear that, yes, there have obviously been some effects, but nothing that's been detrimental to, the future or the progress, which is good. And then kind of talking a little bit more about the fact that you guys are a global company, are there some more successful than not markets that you see? And are there any particular markets that may have been a surprise that are either growing or have been successful for you guys over the years?
Not so much. I mean, the United States is the biggest life sciences market. It's about 2/3 of the world market, maybe 3/4 in some areas, and it certainly plays out for us. It's the most most dynamic market. It's also from a mindset perspective, it's a very competitive market. So if you come with a product that gives a competitive advantage to its users, like ours, like Mary Jo realizing, I use that Blue Washer and my data quality is better. And, maybe one of their competitors isn't using it yet. So just seizing upon an opportunity, while some of the European markets might be a little bit more slow in adopting new things. So the willingness of U.S. scientists and U.S. companies to, frankly, take a bet on a new technology because it gives them an advantage. That's just hugely important for us. And that's, in terms of the other, market sizes or in which country do we have more opportunities or less, it's, there's really no surprises there.
Yeah. I gotcha. And then one other question I'd like to ask is biotech tools industry today. Is there anything that you would change if you had an opportunity, right? Would you change how maybe we do funding or the procurement process, the adoption of new technologies as they come to fruition? Is there anything in that market that might be something that you would change?
Yeah, it's hard to answer. There's not really something where if I had a magic wand, what would I do? I think it's a market that functions fairly well, in particular in the United States as it is. Of course, the funding cuts to NIH and universities that it's going to restrict the pipeline of future technologies. To what extent one has to see, but in general, it's just a very healthy pipeline of innovation coming out of universities and other places being funded, leading to startups, but also just the R&D spending of the big pharmaceutical companies on their own. It's just a juggernaut. I mean, they just really work hard on producing new and better drugs.
One last question. What is the future for BlueCatBio as you continue to grow and produce more amazing technologies in this space? Is this something where you have the eye of somebody who you'd like to be incorporated into the workflow? Would you continue to stay on your own, maybe do some acquisition of your own to have some pipeline and some portfolio fills for yourself? Or do we even know the answer to that question yet?
Yeah, I mean, we sort of, we are bootstrapped, so we have the luxury that we don't really have to think about these things. And we don't really give it all that much thought. So for now, our focus is we have those new products. We have the BlueCombi X, we have the Blue Plate. We have different versions of the Blue Plate. We have the Blue Plate for 96 3D4 and 96 deep wells. There's going to be other versions, just like I mentioned before, for 3D. So our head is so full of things that we want to bring to market. So I think, for the next couple of years, I think we'll just organically grow and really develop the full commercial potential of all of these instruments. Now, having said that, just from a scale perspective, and in particular as we get more into flow cytometry, which is a much bigger market, flow cytometry is a $5 billion market. It's bigger than just drug discovery. So if it makes sense that in the hands of somebody else, it can scale quicker and can create more benefit for a larger set of customers. In particular, if it's bundled with flow cytometers, for instance, we'd certainly look at that, but there's not something that we are sort of actively pursuing.
We'd like to thank our guest, Frank Feist, CEO of BlueCatBio, for joining us on the Biotech Stack. For more information on Frank or BlueCatBio, please check out the links in our show notes below. Thanks for listening, and we'll see you next time.